Good evening. Greetings to one and all gathered here. It gives me an immense warmth and great pleasure to welcome you all to the BioTalk series version 2.4. Last week, our Department of Biotechnology had successfully conducted the BioTalk series version 2.3 on the topic entrepreneurship and career options in biotechnology. Today, in our midst, we have an eminent person, Professor Sunil Khare, who is going to speak on the topic extremophiles as a source of novel biocatalysts. Now, I call upon Ms. Sangeeta, Assistant Professor, to deliver the welcome address.
A pleasant evening to everyone. So, on behalf of St. Joseph College of Engineering Department of Biotechnology, it gives me an immense pleasure to welcome one and all gathered here to attend this Biotalk series version 2.4. So, I extend my kind welcome to Honorable Resource Person Dr. Sunil Kare, who joined with us today to be a part of this program. Good evening, sir. I bid a warm welcome to our beloved Head of the Department, Dr. G. Srikumar sir and Dr. V. Renuga ma'am and my dear colleagues. Last but not the least, I extend my hearty welcome to the pillars of our future India, its my dear students. So we are honored to have you all with us now. I kindly request our Head of the Department, Dr. G. Srikumar sir, to inaugurate the today's session.
Good evening, my dear students and staff members. I'm happy to see you all again for this BioTalk lecture series version 2.4. For the sake of the invited speaker, I wish to highlight some of the credits about our college and the department. St. Joseph College of Engineering is one of the top colleges in India with the 160th position as far as the current NIRF ranking is concerned. It has got 27 years of high reputation in educational society, possessing NBA and NAAC A plus grade accreditations. There are 10 UG programs and 7 PG programs with an affiliation from Anna University, Chennai. Recently, we have received autonomous status also, which will be implemented from the forthcoming academic year. As far as our biotech department is concerned, was started in the year 2002 and having BTech and MTech biotechnology programs. Since inception, we have bagged 139 university ranks with 10 golds and 7 silver medals and maintain 100% pass percentage consistently every year. The department has got sophisticated laboratory facilities and fetched research center recognition from Anna University to do MS by research and PhD programs. We have more than 1.2 crores of government funding for conducting research, and it is actively getting progressed. The effect of current COVID pandemic and subsequent lockdown is worst for the mental health and happiness of the students, where they are unable to meet their friends and enjoy what they used to do during pre-COVID days. The enormous potential that they used to use in their daily life is now completely blocked. And we are somehow continuing the study with the help of different digital platforms such as online classes, teleconferencing, digital open books, online examination, and now the seminar has become webinars, etc. In this crisis, it is imperative to design effective mode of communications for the smooth running of teaching and learning process and also for innovative researching. Understanding the need of the hour, our biotechnology department keeps our students educated towards ongoing research and developments occurring around the world. Our department has the practice of conducting BioTalk lecture series by bringing eminent personalities through webinar and sharing their research views to the entire biotech community that include students, teachers, and research personnels. In this way, today we are delighted to have Dr. Sunil Khare, who is a Dean R&D and Professor of Biochemistry and Enzyme and Microbial Biochemistry Laboratory, Indian Institute of Technology, Delhi, as a resource person. Today's lecture is going to be in the topic of extremophiles as the source of novel biocatalyst, which is highly unique and rare research approach. And I'm sure it is going to be a gateway to our students for innovative research avenues. On behalf of the patrons, Dr. B. Babu Manoharan, Chairman, and Ms. Jessie Priya, Managing Director, and Mr. Sasi Shekhar, Director, and Dr. V.S. Rao, Principal, and Dr. Parvadavardhini, Dean. I welcome the speaker, Professor Sunil Khare, and the biotech staff members, and all the biotech students to this BioTalk webinar series. I also convey my warm felicitation to Dr. G. Bhaskar, Professor, and Madam B. Sangeeta, Assistant Professor, for organizing this wonderful webinar. Now I ask the student coordinator, Mr. Alfred Francis, to give a brief introduction about the invited speaker. All the best to you all. Over to Mr. Alfred.
Thank you, sir.
Yeah.
Professor Sunil Khare, an institute chairperson of biochemistry and currently holds a position Dean R&D at IIT Delhi. He is an alumnus of IIT Delhi and he has received his doctoral degree in biochemistry in 1990. He did his postdoctoral research at National Food Research Institute, Tsukuba, Japan. Professor Khare has more than 30 years of teaching and research experience with 175 plus publications and 3 product process patents, 6 microbial strains, and 50 national GenBank submissions. He has authored 21 book chapters and books and guided more than 15 PhD students. His noteworthy contributions have been in the area of extremophiles, enzyme technology, and nanobiocatalysts, nanotoxicity, food toxicology, and bioremediation. Professor Khare holds a good reputation in the international research community. He has been invited as a visiting professor to the University of Blaise Pascal, Clermont-Ferrand, France in the year 2014 and 18. He was a visiting fellow DBT at Northern Regional Research Laboratory, USDA, Peoria, Illinois, USA. Professor Khare has been honored with awards like United Nations Amway Award in 1998, Malawiya Memorial Award Senior Faculty, BRSI in 2018. He is also an elected fellow of prestigious societies like Royal Society of Chemistry, FRSC, International Forum on Industrial Bioprocesses, France, United Nations University Kirin Fellow, Japan, Fellow of National Academy of Agricultural Sciences, India, and Fellow Biotech Research Society of India, and Fellow Academy of Microbiological Sciences, India. He is also a member fellow National Academy of Sciences, India. He is current Vice President of Biotech Research Society of India. He has also been a member of several professional societies like the American Chemical Society and has served as a chairman member in DST, DBT, CSIR, task force and committees and scientific discovery boards of various universities and institutions. He is the associate editor of Third Biotech BMC Biotechnology, Frontiers in Bioengineering and Biotechnology, and also the review editor of Frontiers in Microbiology and editorial board member of Preparative Biochemistry and Biotechnology, and also the Journal of Environmental Science and Health Part B. Apart from being an academician, he has been actively involved in several administrative responsibilities at Indian Institute of Technology, Delhi. This is reflected by being chairman of GATE JAM IIT Delhi of the year 2016 to 17. Sir, it is an immense pleasure to have you here with us today. I request you to take over the session.
Good afternoon, everyone. I feel humbled by a nice introduction. I'm very thankful to Professor Sri Kumar, the head of the Department of Biotechnology, Professor Bhaskar and Dr. Sangeeta, and other faculty colleagues from the Biotechnology Department of St. Joseph College, and my dear students. Actually, while Professor Sri Kumar was mentioning, I didn't realize that your biotechnology department is almost completing 20 years now. and you have a very good understanding. I have been knowing and meeting your faculty colleagues in different conferences, in different capacities. It's truly impressive. That way, it's my privilege, and I feel greatly honored to share some of the research which we are doing here with you. So what we are doing here is very different kind of research. My main focus has been on the Extremo files. So if you permit, I will start sharing my presentation now. It will take a moment for me. So if my slides are visible on the screen.
No, sir, we can't see your
You
slides.
can't see that? Just a moment. So maybe I made some mistake somewhere. Share. And I will take a moment. You have to share entire screen, so I have to share. All right, now you should be able to see now.
Yes, sir.
Yes, sir, we can.
Now you can see, fine. So I will okay. Thank you. Fine. I think I will go to PPT mode. Okay. So
Yes, sir.
what?
Fine.
Okay. So thank you, and thanks everyone for bearing with me while I was putting the presentation. I'm audible. All right. You are able to listen
Yes,
to me,
sir.
all of you?
Yes, sir.
Okay.
Yes, sir.
So what we are going to discuss today is something called Extremo enzymes or extremozymes. So enzyme word to all the biotechnology students may be a familiar word. These are biocatalysts present in all the kind of cells, perform different kind of reactions, and but what we are going to share is something from extremophiles. So as we all know, philes means loving. So extremophiles are those microorganisms or organisms which love extreme conditions. So if some life is at extreme condition, this should normally be unique and this should have something different than the normal life. And the biocatalyst coming from those organisms which we call extremophiles are called extremoenzymes. So we are going to discuss this today. I can just, maybe someday you will visit this institution. This is Indian Institute of Technology. Our institute was established in 1961. And about 3 years ago, Government of India recognized this institute as Institute of Eminence. We roughly have, so it's a big institute, and we have roughly about 10,000 students, out of which 4,000 may be UG students and about 5,000 plus will be PG students. We have about 600 faculty members as of now, and these members are quite decorated. We have more than 100, decorations where members are fellow of different societies. And on average, this institute publishes about 2,000 research papers per year. And we roughly awarded about 48,000. This old slide, by now we have awarded about 50,000 degrees comprising both UG and PG degree. As far as academic departments are concerned, we have all the engineering major branches we represent. We also have the basic sciences department, physics, chemistry, biology, and we have humanities and management departments. Besides that, IIT has a structure wherein we have certain specified and excellence center of researches. So you will see we have research center in electronics, atmospheric sciences, biomedical engineering, value education, and so on and so forth. Recently, we are also opening some schools actually, which do specialized kind of education and research. So we have the School of Telecommunication, we have a School of IT, and we have a new entity called School of Biological Sciences, Public Policy, etc. I will skip this, but just tell you that we offer both BTech and MTech program and even MSc in the sciences and PhD as well. In BTech, we have both dual degree as well as BTech degree programs. And intake for BTech is through JEE, whereas intake for MTech is through GATE examination. PhD, each department organizes a written test followed by interview. IIT Delhi has collaboration with almost every possible, country in the world. And now I come to the, so I wanted to give you brief exposure of this institution, and I welcome any one of you to visit here. And what you can see here is that your own college, which I was very fascinated to see. So I thought I will put it here, and it's fun for me. I will repeat. So coming back to the talk, enzymes are biocatalysts, and believe me that, of course, this figure is slightly old figure, but total, global market for enzyme is now touching billions of dollars. Some enzymes are more in demand, like proteases, cellulases, etc. But they are very versatile biocatalysts, so they are highly in demand. But what goes against them is that whenever you want to use some enzyme for industrial purpose, condition existing in industrial atmosphere are little harsher. And enzymes, since they come from living cell, they are very labile, they are mild, they are gentle. So if there is an acid or alkaline condition, this denature the protein or enzyme loses the activity. If during the process there is a high salt present, enzyme may lose the activity. If there are solvents present, it may lose the activity. So what industry requires is same as the case with temperature. Industry demand that give us the enzyme which can be stable under our process conditions. That's why people have lots of interest in enzymatic research to develop stable enzymes. And this is being done in past by different kind of methods. So initially at old age time, people used to modify enzyme by putting some chemical. Later on in 70s, people starting immobilizing enzyme. They will attach enzyme to some solid support so that enzyme becomes stable and reusable. Then came the era of enzyme engineering, which you are studying currently, and site-directed mutagenesis. Herein, what you can do is you can change certain amino acids in the enzyme by molecular techniques, and it becomes stable. You have to see how you can stabilize it. Directed evolution is kind of DNA reshuffling, which also makes it, Our approach has been to screen the enzymes from extremophiles. As I said in the beginning, extremophiles are microorganisms which live under extreme condition. And we believe that if they live under the extreme conditions, their enzyme should also be able to perform the reactions under extreme conditions. Just to make you better understand it, you can see some picture and you can understand. Here also you find microorganisms. So how these microorganisms are surviving? Because their enzymes are tough, they work under extreme condition, and that's why these microorganisms are able to perform all their functions under extreme condition. So that in this way, they have the nature's clue of enzyme stability. So you also find enzymes at a very high temperature. You find microorganisms, thermophiles, at very high temperature. You call them hyperthermophile. Now, the BTech may not be so, but MTech and PhD students may be using PCR, and you can understand that PCR was invented only after it was discovered that there is an enzyme which can be stable under PCR condition. So that enzyme is Taq polymerase. I think you all may be understanding that enzyme that comes from Thermus aquaticus and that can sustain itself at 90 degrees. Beyond that, even it was observed that there is an organism called Pyrococcus. In Japan, people found out, and you can boil it actually. Such microorganisms have enzymes which are very stable under high temperature. Similarly, at low temperature, how many of you can believe this, that even under this kind of mountain range full of snow, microorganisms are there, even in Antarctica. And one microorganism which was isolated from a very famous, from Candida, from Antarctica, it is called Candida antarctica, is the name of microbes. And its lipase is very, very useful in the industry because it is stable at very low temperature. Similarly, you can think of acidophiles, wherein the organism is stable under acidic condition. So you can see the acidothiobacillus, alkaline conditions, Bacillus pseudoformis. And then I will come to a very favorite topic of mine called halophiles, wherein microorganisms survive under high salt. So you are actually surrounded by sea coast, and sea coast means sea water itself is highly salty. And if it contains, let's say, 2 molar or 3 molar, sometimes 5 molar salt, how many of you would believe that in sea water there are microorganisms surviving? They do. They are called halophiles, halobacteria, haloarchaea species. And what is interesting is that they do need more than at least minimum 2 molar salt for their growth. If you don't give them salt, they will not survive. So this is very interesting set of schemophiles. And you will be you can sometimes collect the seawater from Marina Beach or somewhere. And then you just try to culture them. If you want to culture them, you have to give high salt. Otherwise, you can't culture them. And same thing repeated in a different manner. What I'm trying to tell you here is that you all know I think you may be familiar with the word called protein data bank or NCBI or gene banks, wherein you will see that about 3.3 million protein sequences have been submitted, but only 8% corresponds to extremophiles. So that means we need to work more and find out more and more. Okay. So I will come back now to halophiles and focus more on halophile here. And so this is some of the conditions where the halophile survive. This could be seawater, it could be saltern. You understand saltern. Saltern means a soil or salt pan where water has stayed for a long time. Then it evaporates and you'll find concentrated brine actually. Even there you find the halophiles, salt lakes, etc. Even saline soil actually will have these microorganisms. You can call them halophiles. Now, just to give you a brief thing, okay, just to give you a brief account, if you have idea about a cell, so if you put a cell in the salt solution, cell will shrivel actually, shrink. But if halophile are living in the salt water, why are they not shriveling? So that is the reason that they are there in presence of salt, but they have equivalent salt inside their cytoplasm. That's why this osmotic effect is not there. So The second question, which I already initiated, is that then how do they survive in the salt? So it is now known that they have two kind of strategies to cope up with high salt around the cells. One is called compatible solute strategy. In this case, the cells do not have matching salt, but they have certain molecules which you call halolytes, okay, or osmolytes. These are something like betaines, actoines, polyols. They are in plenty in the cytoplasm of these cells, and they maintain the cellular environment and sustain the cell under high salt. Another strategy is salting in strategy, which is simple, wherein if outside salt is 1 molar, cell will also have 1 molar salt and cell will survive. So here cell has matching salt concentration in the inside the cytoplasm. So now coming back to enzyme, actually, so what is observed is that enzymes which have been sourced from halophiles are active at high salt concentration in the range of 0.2 to 5.2 molar. So you call it moderately halophile and extremely halophile kind of organisms. Now they are very useful in detergent, where because these enzymes are used in detergent because their salt is present and this enzyme survives in the salt. Second is that they are better catalysts for synthesis actually. So synthesis, please just to brush up your basic concepts. If you carry out reaction of enzymatic or any reaction in forward direction, you need water and it is called hydrolysis. If you remove water, the reverse reaction will take place. So reverse reaction means synthesis. For example, if there are two monosaccharides, you put in water. If there is a disaccharide, let's say lactose, you put in water and put the enzyme, it will form glucose and galactose. That is hydrolysis. If you remove the water, glucose and galactose will join together to form the lactose again. That is synthesis, the reverse reaction. So these can be used for synthesis. So that's why other application which people perceive about halophiles are hypersaline waste. So some waste are having high salt. For their treatment, these are good systems. And lastly, we are also interested to understand their structure and function. How do they perform their function? So now I will narrate the problems which normally are faced by people working in this area. First is, you should search as many halophiles as possible for finding out novel enzymes. Question is, do they possess some unique property which are not present in non-halophiles that you should see each time? So that is first question. Screen them and find out what are those unique properties which is not reported before. Second is, if they have certain unique enzymes which we can call halophilic enzymes or salt-loving enzymes. Now salt-loving enzymes means whenever you have to catalyze their reaction or you want to keep them in test tube, you should give high salt in the test tube, otherwise they will get denatured. This is a problem. If I want to purify such enzyme in my chromatography setup or wherever I am trying to purify, you understand purification means removing one particular enzyme or protein from the mixture of protein and enzymes. This is done by chromatography. So there are, molecular sieve, there are ion exchange chromatography and different kinds of chromatographies are there. In each chromatography, salt has not to be there because otherwise chromatography gets disturbed. But if salt is not there, then these enzymes will be denatured. So purification of halophilic enzyme is a challenge. Thirdly, we would like to understand what is difference in their structure and genes and molecular architecture so that they are stable in salt and low water stability. Why I say low water? When there is a high salt, there is always low water. And then lastly, can we exploit these enzymes for industrial application? Initially, I told you that in the industry, you need enzymes which are stable. So the question is, can these be good biocatalysts for industrial applications? So first is searching for new halophiles. I think I can tell you that my students went to different parts of India for collecting samples. So you can see some of these are like Goa is a good place where you Sambar Lake is a lake, salt lake in Rajasthan. Even you are maybe familiar with brackish waters and Kozhikode, etc. And we also went to other Gujarat also. Gujarat coast also we have collected samples. So you will, what I'm trying to show you here is that on the first column, you will see the site from where we collected the sample. The second column will show you how much salt is required for growth of organism which we isolated. Third is name which we gave to them, or we matched it with them database. Next column is gene bank accession number. Then we deposit some bacteria to gene bank. Gene of that bacteria to gene bank, we get a number. And you may be familiar that microorganisms can be phylogenetically related to other microorganisms, and then how much similarity is there. So what I will read first, Sambar Lake, which is a salt lake in the Rajasthan, and you will see that most of the microorganisms which we isolated from there need salt from 10 to 15%. Their accession number is all right, but they are related to Geomicrobium, Staphylococcus, Bullellia. Next is Kozhikode. I will just give this example because you are familiar with this. Here, the salt requirement was 5%, and we mostly found Marinobacter and Virgi bacillus, etc. Now I will move. So this way we collected from Gujarat and Somnath and Veraval, and we found different kind of, micro halophiles. Actually, they are different, and we deposited them. And just to sum it up, the major halophiles which we found along Indian coastal lines are Chromohalobacter, Halomonas, Marinobacter, Halobacillus, Virgi bacillus, Ocinobacillus, and Geomicrobium. These are major genera we found, and different species, of course. Then we actually screened them, and they have very good proteases, very interesting lipases and amylases. Okay. Since our interest was in enzymes, so we saw for each microorganism or halophile, we saw the enzyme, and then so in this table you will see what we have done is that name of microorganism, then the enzyme which was the enzyme which was present in highest amount in that organism, then production level of that enzyme, pH optima of that enzyme. pH optima means at what pH they are maximally active, and how much salt this enzyme needs for biocatalysis, at what temperature they can perform the activity, and last is thermal stability, and in between you will see solvent stability. I have initially told you, just to reiterate, that when some organisms or protein is coming from high salt, water is low. Low water means in mimicking the organic solvents, hexane, decane, dodecane, these are low water solvents, or we call organic solvents. So we wanted to see whether, since they live in high salt, low water, are they stable in organic solvent? I will tell you later why this is important. So now I will just read one, first 4 columns or one. The Sambar Lake gave us proteases, amylases, which has pH optima in alkaline range, salt optima and they do need salt more than 3% all the time. They are active at little higher temperature, 50 degree or so, and they are stable in organic solvent. So was the case in most of the cases. Okay. So I will not. So again here same thing, from different locations, different organisms, different enzymes, their alkaline pH, the thermal stability, and their stability in organic solvent. So I can sum up by telling that the enzymes which we isolated from halophiles were alkaline. They were working mostly at alkaline pH. They were stable and do require high salt, and they were stable in organic solvents. I have already told you, because salt reduces the water activity, so it's like low water, and so they can function in organic solvent. Solvent stable enzymes are very unique and in high demand in the industry for synthesis. For instance, I can tell you one product which comes by synthesis is aspartame. Many of you may be having your relatives, friends, or some acquaintance which are diabetic, and then they take not sugar but they take aspartame tablet. This is a dipeptide, but to create this dipeptide you need to carry out the reverse reaction or synthetic reaction. Industry does that, and they use thermolysine, which is stable in organic solvent. So organic solvent stable enzymes are high in demand by the industry for synthesis of oligosaccharides, peptides, and lipids of different kind. This is elaborated here. Since students are there, I would like to tell you that look at this reaction. Here is AB is the substrate. If aqueous media is there, hydrolysis will take place and AB will split into A plus B. This is product. But if these two products are there, A plus B, and I carry out the reaction in organic solvent, then this A plus B will form AB. This is synthesis. And to do this, you don't need aqueous media. You need non-aqueous media. What people use, this is called, please understand term, which is very common. There is a research lab in MIT called non-aqueous enzymology research lab, where they carry out all the enzymology in organic solvent. So here organic solvent means hexane, heptane. So if you put hexane here, what you will see is, I will take 1 minute. Sorry. So you need enzyme but for doing this, you need an enzyme which is stable in organic solvents. Normally what happens, many of you may be knowing protein structure. Okay, so the protein is a tertiary three-dimensional structure, but if you put protein in hexane, protein will denature. In protein, typically the hydrophobic groups or hydrophobic amino acids, which are mainly leucine, isoleucine, valine, etc., they are buried inside the cavity, and hydrophilic amino acids like you see serine and aspartic acid, lysine, etc., they are on the surface. So the moment you put this protein or enzyme in organic solvent, the buried hydrophobic groups they come out, okay, and protein gets open, and once it is denatured, it cannot perform the catalysis. So such protein which are stable in hexane or organic solvent are very rare, and they are very useful for synthesis. That's why people are interested in having the enzymes which can be stable in the organic solvent, especially all the lipolytic reaction, because lipase is actually the substrate for lipase are oil. Oil can be dissolved only in organic solvent, so you need enzymes which are stable in organic solvent. So there are many such examples, but carry home message for you is that solvent stable enzymes are very important for industrial applications. Now, second question which I was asking was how to purify these halophilic enzymes. If they are solvent stable, they are fantastic. So my previous message was halophiles are unique organisms which are stable in high salt. Their enzymes are further unique. They are alkaline and they are stable in organic solvents. Now, having said that, my next question would be that can we actually purify them? Because I told you that for purifying them, it is difficult task. Okay, so how to do that? For this, we tried few things. So I'm giving you one example. Just. Just 1 minute. I need the mouse. Okay. So I'm giving you one example of geomicrobium. This was isolated from Sambar Lake. It was, so you can see the cells, you can see single cell. K is not Khare. This is my student, his name was Karan. So he was very fond of this. So if you have to see protease activity on agar plate, basically what you can do is you put the gelatin here, inoculate the media, the microorganism, you will see the zone of hydrolysis, because if it is a protease producer, I think students may understand. If I put gelatin, it's a protein. And if my organism uses protein, it will create a hello zone. So that means it is a protease producer because it is utilizing. So it is a protease producer. We submitted this to Chandigarh, where this called microbial type culture collection. This organism is available there in this number. I will just take 1 minute. How to bring out the cursor in this? Sorry, what I wanted was that can I have a cursor here, but I could not find the cursor. Sorry. Okay, now I'm fine. So sorry for this inconvenience, but I think it's better now. So you can see this, that we are purifying the, what we did simply, we tried a technique called hydrophobic interaction chromatography. In this case, we believe that since the enzyme from halophiles are stable in organic solvent, their surface should have hydrophobic amino acids. That's what my faith was. And if that is so, then if I have a chromatographic column, which is based on hydrophobic matrix, phenyl cephaloz is phenyl is hydrophobic. So if I have this culture and I did ultrafiltration, that means I removed the scum, then I took the retentate where protease activity was there, and I put it in the hydrophobic interaction column. What will happen, you see here, other protein which are non-hydrophobic will come out, just elute out, wash out. The protein which is hydrophobic will bound to the column, and when I give ethylene glycol, which is much more hydrophobic, it will come out. So we could get single peak and with the activity, and we could purify this enzyme by hydrophobic interaction chromatography by 22-fold and pure enzyme. This technique looks very good technique for purification of solvent stable halophilic enzymes. And we could show then that it's a single band showing pure enzyme. Zymography, you understand, this is activity staining. That means whatever we have purified is protease only. So we could actually. Second is another similar way. So I will go quickly now. So the amylase from some Koji code, and here it is starch agar plate, hydrolysis zone shows amylase production. So this is again halophile, we call it marinobacteria. And here again we could purify. And then another Virgi bacillus from some other location, and it's a protease producer. We can again purify it, and another bacillus we could again purify, and then another lipase enzyme we could again purify it. So what I wanted to tell you that though HIC looks fine, some of this could be purified by different techniques also. But whenever we purified them by some other chromatography, we added salt all the time as soon as enzyme came out. So this was difficult actually. But I would like to say that hydrophobic interaction chromatography seems to be a good technique for purification of halophilic enzyme. Now, I will just simply summarize the characteristics of the halophilic enzyme which we purified. You would see their molecular mass is in this range. They are not big proteins, they are small proteins. Their pH optima is alkaline, you will see. Their temperature optima is 50 plus all the time. They are stable at high temperature. Their salt optima means how much salt you give, so they are most active. 1% salt is good for them, and then they are stable in detergent. I will come to that point later. And then they have KM, Remax, etc. And they are mostly serine proteins. Okay. So now the last question and last part is, how are they stable? What is the molecular basis of their stability in the salt and low water or organic solvent? So you please understand, they are not only stable in the salt. Salt means NaCl. They are also stable at low water and organic solvent. So this is a typical way to do that. I think we can just move here. I can explain what it is. So we actually took out, let's say, protease gene, and then we ligated it, cloned and transformed, and then again we picked up the white colonies where we got the cloned protease, and then we made a restriction cut here, so we got the plasmid, and we could release the insert here, which was about the size of protease which we actually cloned. So what we can do is we could isolate this protease gene and purify this. This gene, when we sequenced, I think you will see, probably I missed some points. So we took out that gene, and then based on gene, we deduced the amino acid composition. What you'll find here, this protein, the protease, which is halophilic, is having very high amount of hydrophobic amino, much higher than hydrophilic. Okay. So this means what I'm trying to emphasize here is that the proteins or enzymes from halophiles are hydrophobic in nature. And when we submitted this sequence to a server, which is K2D2 server in Canada, and also we submitted, that ITSR server, what we found is structure is like this. So this protein, which we discussed so far, this protease of geomicrobial, and this is geomicrobial protease, and when we saw its model, it's a tertiary structure, three-dimensional structure, this yellow thing is hydrophobic surface. Now you will be able to appreciate that these halophilic protease are very, very hydrophobic in surface. So this hydrophobic surface gives them solvent stability. Other thing which you can see here is, why are they stable in salts? They do need NaCl. So you will see these are actually polar amino acid, okay? Charged amino acid, aspartic acid, lysine. So charged amino acid are also there. So what is there? Not only their surface is hydrophobic, their surface also has patches of charged amino acid. So that's why they are stable in salt. This kind of characteristics makes them stable in organic solvent and salt. This is salt optima of that protease. So 1% salt gives maximum activity. Now what I am trying to show here in this slide, if you take these protease, so control is this. This is control. This is control. That means in buffer. This protease is taken in buffer and kept in buffer for 12, 24, 36 and up to 72 hours. It's stable in buffer. But if you keep that in benzene, this is still stable up to 72 hours. You keep it in heptane, this is still stable. You keep, so whatever organic solvents you can think of except butanol, which is a polar one. I will tell you later why. Polar are bad because they strip off the water molecule. So polar solvents are not good for solvent stabilization. But other than that, you see most of the organic solvents at high concentration. It was 25% solvent and they are stable. Even we went up to 75%, 90% stable. So a carry home message here is that they are need salt, they are stable at organic solvent. This is because they have this structure, and those little bit chemistry, I am sorry, please bear with me. I want to show you why NaCl is important for these proteins. We say they are living in the sea, the organisms are under salt. Enzymes also need salt. They are stable in salt. Now why? So if you have some idea, protein have 4 levels of structure. Primary structure, which is simple amino acid sequence. Proteins have secondary structure, which means either they are alpha helix or beta sheet. Tertiary structure means they are globular structures, and quaternary means 2 globules coming together. This CD spectra, CD circular dichroism, gives an idea about secondary structure of the protein. So typically, if you remove salt from protease, there is no structure at all. It is open chain, primary structure only. If then I add 1% salt in this denatured, no salt here, I added 1% NaCl, it start gaining the structure. I added 2%, more structure. I added 5%, more structure. 10%, perfect alpha helix. So you see, to maintain the structure, this halophilic protease or protein needs NaCl. If you remove NaCl, structure is lost. Same we saw in presence of organic solvents. If NaCl is present, these are organic solvents. So this is all sort of organic solvents and no NaCl. Enzyme has no stability, structure is lost in organic solvent. But if I put 1% NaCl and then add these organic solvents, it acquires structure. So the message is that NaCl exerts a protective effect to maintain the structure even in presence of organic solvent. So what I was saying in the beginning, they are stable in salt, but this salt is very necessary. They are stable in organic solvent, but salt is still necessary to give them this stability in organic solvent. Similar thing we observed in case of amylase. Again here they are stable in organic solvent. The surface of this Marinobacter amylase is again similar kind. We did the tertiary structure here. So if salt is removed, structure is lost. Without salt, this is open. Without salt, tertiary structure is reduced. But if you give salt, structure is regained. Similarly, if you give salt and give hexane, ethanol or helicane, structure is regained. So again, it confirms that NaCl is necessary as a protective agent, and then it is stable in organic solvent. Similarly, another protease, CD, shows the same thing. Another protease, CD lipase protease, it shows same thing. So the conclusion is that the surface of such enzymes are charged and hydrophobic. That's why they are stable in salt and organic solvent. And then their secondary structure is stable in presence of solvent. However, salt is necessary. Salt is necessary for their secondary and tertiary structure both. So now last part is that can we use them for industry? Okay. And I will just take 5 minutes. I know it has been pretty long for you. How many of you have used detergent in the laundry at home? So earlier time when our grandmothers used to do it, they will put hot water, salt and everything and then wash it. Now you have two kind of detergent. Ones are very expensive detergent, wherein you say all kind of stains are gone. Like your oil stain, your protein stains are gone. But these are expensive detergents. And there are some cheap detergent. So stain is not removed. What is happening is under these stains, in these detergents, enzymes are added actually. So those enzymes which are present, like suppose lipase is present or protease is present, then they remove the stains. So what this experiment is showing, that we take simple Surf Excel, Ariel, Tide, Rain and Wheel detergent, add protease here for 1 hour, 2 hour and 3 hour, and then see the stability of enzyme. So we see that protease in 1 hour is quite stable, 2 hours slight loss, 3 hours in presence of detergent. Now you can ask me, why are they losing activity in presence of detergent? Detergent have lots many things. So that's why enzyme gets denatured. But these protease from halophile, they are still stable up to 2 hours. And 2 hours is roughly the time for washing. Okay? And now I will show you what. This is a blood-stained fabric. Okay? So we took a blood-stained fabric, incubated with 5 ml detergent. Simple. And then we see some removal is there, but stain is there. But in second case, we took fabric, stained it, and incubated with enzyme and detergent. You see it's much cleaner now. So this all remaining. So this enzyme is removing blood proteins. So this halophilic enzyme was very good for detergent application. Amylase we used for synthetic reaction. We could make maltose, maltotriose and maltotetrose, which is very difficult. So you drink these days the Yakult, you drink probiotics, they have oligosaccharides, which can be synthesized by solvent stable amylases. And another application of another protease, you see the stains are gone. And this is without enzyme. So the conclusion is that they can be good for detergent application. They can be good for oligosaccharide synthesis and other kind of synthesis. Overall, I would like to conclude that halophiles are very good source of alkaline. Alkaline because detergents are alkaline, remember. So that's why these, I will go back, tell you that in detergent, you need alkaline thing, you need slightly higher temperature, and you need salt. These enzymes are stable in alkali, which means detergent, salt of the detergent, and solvent is stable. So these halophiles could be good source of very novel kind of enzyme, wherein they have this kind of properties. These properties make them useful for synthesis because in organic solvent. They make them useful for detergent application. So I think halophiles, in my opinion, are vast resource, is still untapped. Lots of research can be done on this. I would like to acknowledge my students, some PhD students who have done work on this. Different enzyme, different student, and the fund was supported by DBT. And this is the group which function. We teachers are teachers. We just tell them, guide them, facilitate them, but these are actual hands who does all this. So thanks to them, and thanks to all of you for giving a very patience hearing to me for this long time. If you have any question, I will be happy to take it forward. Thank you very much.
Thank you, sir. Thank you, sir, for this wonderful lecture and enlightening us on the topic extremophiles as a source of novel biocatalysts. So actually, we have a question.
Yeah.
Uh, the recently discovered rotifers are 24,000 years old from a permafrost. What would be the reason for the survival of the organism after so many years?
Yeah, I read that paper also, and actually, it is having those molecules, which remain frozen actually, but they were able to perform minimum metabolic activity, and kind of it's kind of, you must have heard hibernation concept, right? It's a hibernation, very minimum energy, but they were able to use certain molecules at a very low temperature. Very good question. I feel impressed. Lots more to be known about it, but this is my impression about it.
Yes, sir. So I guess that's all the questions being asked in the chat box, sir. So like, I kindly request Dr. Bhaskar, sir, to give the vote of thanks.
Yes, Albert, 1 minute. So, good evening to one and all. On behalf of the Department of Biotechnology, St. Joseph's College of Engineering, and on my own behalf, I take this opportunity to propose vote of thanks. I thank our today's chief guest and resource person, Professor Sunil Kare, Director R&D IIT Delhi, for his talk on external as a source of novel biocatalyst. We are really united with your presence and the knowledge shared on extremo enzymes and their applications, sir. So thank you for your very interesting and thought-provoking presentation, sir. Thank you, sir. I express my uh, profound gratitude to our college management team, chairman, managing director, director for their constant motivation and support for organizing this kind of events. I would like to thank our principal and the dean for their continuous support. I am very grateful to our HODs for giving us an opportunity to organize these kind of events for the betterment of our students. A special thanks to our faculty members and the organizing team, Alfred and others for their timely support and coordination. Our heartfelt thanks to our students for their active participation. Once again, I thank our chief guest, Professor Sunil Karesa, for accepting our invitation and spending his valuable time and sharing his knowledge on external fields. With these warm words and a kind message, we move to the end of today's talk. Thank you, sir. Thank you very much
So
for your.
I also thank all of you, and it was pleasure for me to interact with all of you. And thank you,
Yes, sir. Yes, sir. Thank you. Thank you, sir.
And other faculty colleagues. Thank you very much.
Thank you, sir. Thank you very much. Thank you, sir. Over to Alfred.
Once again, I thank all the participants for attending this event. We will meet you soon with another BioTalk series in the upcoming weeks. Thank you.
Thank you, sir. Thank you.